Alright, this video is gonna be about microarrays, and so both southern blot and northern blot use a fluid phase single probe and immobilized complex targets. So you have the target on that membrane we saw yesterday. So the target's on this membrane over here, and so the target is fixed, and then you have the probe that's floating around in the solution. So these are the probes which are floating around the solution. That's the same case for northern and southern blot. And in microarrays, what you have is you have a fixed probe and you have many different targets that are floating around, and this is going to come in attached to them. And with microarrays, you can test for many different targets at the same time. And then these expression arrays are replacing northern blots, and the genomic arrays are replacing the southern blots. For southern blots are used for single nucleotide polymorphisms and copy number variants. So like an A is changed to a T, and if you want to detect that, you can use a genomic array. And then the arrays is also the arrays you can also do sequencing with them. So this is basically how a microarray works. So you have the fixed probes on glass or different kind of binding structures, and then you have the probes, and then you have the labeled target, the fluorescence labeled target. It's going to come and bind. So if it's bind partially, it's going to be weak. It's going to be a weak bond. So what you do is you isolate the mRNA from two different cells, and this is in two different developmental stages. And you isolate the mRNA, and then you use reverse transcriptase, but you use fluorescently labeled deoxyribonucleotide triphosphates. So then this will glow green, that'll be red. And then you have the microarray tray here, and then you have on each one of these you have a different probe, and these are fluorescent, so they'll bind to these trays, and then you'll see these green and red dots. So over here we were looking for a specific gene over here, and that specific gene was found in this one, so it's green. And we were looking for a specific one over here. That specific one was found in the red one over here. Over here it's yellow, so it was found in both cells, so they might be housekeeping genes. And this is a clinical application of the microarray MammaPrint, where you have probes for 70 different genes that are involved in cancer. So some of the genes might be that have to be there, or some of the genes that are like mutated and not supposed to be there. So can analyze all those genes in a 70 of those genes on a microarray at the same time. And so if you know if the patient has a lot of those 70 genes, then you might recommend them for chemotherapy. If he's not matching up for all 70, maybe if she's only matching for like 2 or 3 of those genes, maybe you wouldn't do chemotherapy, rather just do hormone therapy. And so you put the 70 genes probes over here, and then you add the patient's DNA and see which ones light up, and then based on the results you continue with the treatment. Right, that's it for microarrays.